mutation nomenclature, some additional rules. There is no nucleotide 0 while describing the variants on the gene level. Nucleotide 1 is actually the first nucleotide of ATG. ATG is the first codon that is also called as initiation codon. So first alphabet, that is adenine, is the first nucleotide. The nucleotides at the 5 end of ATG translation, they are numbered in the form of -1, -2, -3, -4. If we take the example of a gene, this is the sequence of the gene, and here is ATG. So this A is actually nucleotide 1, 2, 3 and so on. While on the other hand, if there are nucleotides here, that can be, let's say, T, C, G. So this nucleotide, that is T, it will be written as -1, then -2, -3 and -4. So the first nucleotide of the ATG is nucleotide 1, while all the nucleotides which are present on the 5 end, take this one as 5 end of the gene and this one as 3 end of the gene. So those nucleotides which are present towards the 5 end, they will be marked as -1, -2, -3, while those nucleotides which are towards the 3 end, they will be marked as 1, 2, 3, 4 and so on. The nucleotide at the 3 end of the translation is stop codon. Once there is a stop codon, the next nucleotide will be marked with steric. If we see here the structure of the gene, this is a sequence of the gene. Here is ATG, this is ATG, this is 5 end of the gene, this is 3 end of the gene, and here is the stop codon. If here is stop codon, then the next nucleotide, that is here, it will be marked by 1 and steric. The next nucleotide will be marked as 2 and steric. The next nucleotide will be marked as 3 and steric. So all the nucleotides which are present beyond the stop codon, they will be marked by 1 steric, 2 steric, 3 steric, in this way. Nucleotide numbering is purely arbitrary. It starts from 1. There is no nucleotide 0, as we have seen in the sequence of the gene, that if gene starts from here towards this direction, towards the 3 end, it will start by 1, 2, 3, 4, 5, 6, while on the other hand, it starts as -1, -2, -3, -4, -5. So there is no nucleotide 0 in the sequence of the DNA, and usually no sign of plus and minus used. The sequence should include all the nucleotides covering the entire sequence of the gene, starting from 5 end up to 3 end. So while describing the sequence variants, either at the level of DNA, RNA or protein, all the instructions that have been formulated by HGNC, they should be considered.